Okay, this is the second part of the blood. We're going to be concentrating mostly on the white blood cells as well as how to do the blood typing. So let me begin by talk a little bit about the leukocytes. Leukocytes, another name for it is the white blood cells. All right. Um, the white blood cells or leukocytes, you might have heard about leukemia, which is a cancer of white blood cells. They come in different shapes and different forms. All right. They're much less numerous than the red blood cells. So they make up only up to 1% of the total blood volume. What that means is they're very, very little in terms of their numbers. And however, if we get the infection, yes, their number can rise up. Depends on the type of infection or the type of problem that we might have. Can leave the capillary via diapedesis. So remember, the capillaries, they have really, really thin cells. All right. So if there is a white blood cells in here, okay, first they can adhere and then they could leave. So let's show one that is leaving, right? See, they're leaving, all right? Uh, Now, maybe this is the skin, maybe, your skin, a nail went in there, and there are microbes that are there. So these white blood cells, they move through the tissue. That's what it's saying. This is called diapedesis. And then they engulf these microorganisms, bacteria, and things like that. All right. So interestingly enough, they respond to the bacteria, viral invasion, even protozoa, chemicals. It really depends. Parasites. They are the defenders of the body, obviously. Now there are different type of defenders. One type is called granulocytes. Granulocytes, they have granules, all right? These are as follows: neutrophil, eosinophils, and basophil. So the granules stain with certain type of coloring. It's known as a right stain. Don't worry too much about it. Usually they don't live that long. I mean, red blood cells, they live like 100 to 120 days. These are very short-lived. And I tell you, in particular ones, you don't need to memorize, but you could go ahead and take a look at. Their nucleus is not like, general, like a generalized cell. Some of them, they have lobes, and they're all pretty much phagocytic cells. That means they could eat other things, basically, microbes, bacteria, protozoa, things in that nature. All right, so let's go ahead and take a look at the first one, neutrophil. Neutrophil are the most common type of the leukocytes. They're the most abundant type when you're looking at a different type of leukocytes. All right. Uh, they are known as bacteria slayers. They have hydraulic enzymes, and when they have, engulf the bacteria and whatnot, they can take care of it. All right, peroxidases, hydraulic enzyme, and certain other type of a protein. They're all help them to kind of like digest stuff. Neutrophil. Let's go ahead and take a look at what the neutrophil looks like so you'll know the shape too. All right, so taking a look at the neutrophil, you see there are lobe nuclei right here that you probably seen it in anatomy. In this case, it's three. It just have one nucleus, but they're just like with the thread, they are connected to one another. That's why it's called lobe nuclei. So take a look right here, right there. Okay, they're multi-lobed. All right. So their development lasts about 6 to 9 days, and their life expectancy (LS) is 6 hours to few days. They phagocytize bacteria, so it tells you what they do. All right, you could have up to maybe about 75% or so neutrophil. Obviously, if you're infected with bacteria, that numbers will go up because they manufacture more. Next up, you have eosinophil. Eosinophils, they are distinguished by these red dots, and they're usually bilobed nuclei. All right, they're much lesser than the neutrophil. They're there for parasitic worm infection. They can also play a role in chemical and allergy in terms of the inflammation. Again, life expectancy, uh, 8 to 12 days. Don't dwell on these numbers. They could go a little bit up and down. Same with the numbers. All right. So, depending on which textbook you read, they might have a slightly different range. So, pay attention to that. All right. But generally speaking, you could have maybe up to right around, I would say, 4% eosinophil, but usually about 2 to 4%. Next up, you go to the last type of granulocytes. And again, look at see the granules. You can see the granules, they're darker blue, right? And in here, eosinophil, they're a little bit red. All right, so let's go ahead. And the last one is called the basophil. Basophil are the rarest type. You have like maybe less than 1% of the white blood cell. That means out of 100 white blood cells that you count, you'd be lucky if you encounter one of them. All right, so they're the least numerous one. All right, so again, they're releasing histamine for inflammation. They contain heparin and also anticoagulant. All right. Then we go to the agranulocytes. Agranulocytes, you don't see any granules. These that stain darker are called granules. These you could see there's not much granules absorbed in the cytoplasm. So the lymphocytes are the least, I'm sorry, they're the smallest type of white blood cells in terms of size. Okay, so it could last up to months sometimes. There are two kinds of lymphocytes. Uh, you might have heard about T lymphocytes or T cells and B lymphocytes or B cells. B cells, they produce the antibody. T cells, they attack the infected cells or cancerous cells. T cells, they mature in the thymus. B cells, they come straight from the bone marrow. You cannot distinguish the two, but most of the cytoplasm is occupied by the nucleus, as you can see. They're really small, and again, depending on the which textbook you read, but you could have about maybe 25 to 40% of the white blood cells being the lymphocytes. All right. So uh, again, they can destroy the infected cells, cancer cells. Most importantly, the B cells, they produce antibodies. Next up, we go to the largest type, and that will be called monocytes. Monocytes are the largest type. You would have up to about 8% or so of the white blood cells, monocytes. Monocytes, not only they're large, but they could be quite easily distinguishable because they usually have a U-shaped nucleus, right? It looks like a horseshoe or a kidney bean. And they're major macrophages too. So they engulf and destroy, whether it's chemicals, whether it's protozoas, malarias, things in that nature. All right. So then you'll have platelets, which is fragmented cells, and you can see, and that's how it looks like for blood clotting. So you have the function on the very right, all right, some of the description, what do they look like, and so on, how long is going to last, and what amount of them are available, all right. But you could just remember this sentence if you want, not let monkeys eat banana, and NOT is for neutrophil, that's the most one. L for lymphocytes, second most numerous one. M for monocytes. Monocytes then is after that. So EAT for eosinophils and B for banana for the basophil. All right. So that's how you could go from the most to the least. All right, neutrophil is the most, all right, in terms of the numbers, and the basophil are the least in terms of the numbers, and everything else is just follows. So if you want to know the order, that might be easy way to recognize this. All right, next up, we're going to go ahead and take a look at the blood grouping. In general, if you're looking at the type of a blood and looking at, you know, to see, for instance, there is really very high number of neutrophil, that might tell you something. It might tell you probably that person has bacterial infection. All right. If it has parasitic worm infection, most likely the number of eosinophil is going to go up. Eosinophilia, all right, and so on. So you get the idea. These are kind of like a defenders, but there are different type of enemy. Just like a country have different defense system, the Navy take care of the ocean, Air Force taking of the air, Marine taking of the land. There is a counter-terrorist group that deals with the terrorist activity. So everybody, the defense of the system of the country has all these forces. Nonetheless, sometimes, when you look at other countries, they turn against the people and they do us harm. Similarly, in here, autoimmune diseases are like that. They turn against the body cells and they start attacking the body cells. All right, like MS that we discussed when the myelinated sheets were getting destroyed. All right, so those are autoimmune reactions. Automatically, our immune system is attacking its own cells. All right, not recognize them as friendly. Anyways, the immunology is a separated lecture by itself, but right now we are going to go to the ABO blood system. All right. Uh, this, again, for anything that I left behind, you can go ahead and take a look at the simulation, help you understand, but I wanted you guys to listen to this one first, if you can, and then take an attempt on the ABO simulation model or antibodies. As you know, there are different antigen on the surface of our red blood cells. You could have A antigen or a B antigen or both of them, A B antigen or none, no antigen. All right. In terms of the antigen, there is also a D antigen that usually we refer it to the RH antigen, which signifies and allow us to know whether it is a positive or a negative. All right. Uh, Also, in the plasma portion of the blood, there might be antibodies. All right. So, upon getting a wrong blood or different microorganism in the blood, the body produces antibodies. Again, this is primarily the job of the B cells, also as a plasma cells as well. They produce different antibodies. All right. So they could have anti-A or anti-B. Anti means against. So if the person is B blood type, you give them an A blood type, they're gonna have anti-A production going on because they want to attack the A. That's a wrong blood type. They cannot recognize the ID card or the antigen on the surface. Similar with anti-B. If the person is A, you can't give them a B because then, you know, they have antibody B. They will against the B antigen, revoke against the B antigen. There's also anti-D or anti-RH. All right. So you've seen the genetics of the ABO. I'll go over it shortly again. But first, I would like to go ahead and talk a little bit about what happens if you give the person the wrong blood. Agglutination happens. Agglutination or agglutination is referred to the clumping. All right, the red blood cells, they clump together via the antibodies, and pretty soon the body reacts to them. They try to destroy them, and this is usually known as a hemolytic reaction, hemoblood lytic reaction. Okay, so that's the idea, but let's kind of like go through it a little bit further to understand this. All right. In this particular figure, they have the A antigen on the surface as a triangle and B antigen as a circle, and they have a AB. So this must be a AB group because it has both of these antigen. All right. B antigen, it has only B. B blood type, they have only B antigen. A blood type, they have only A. And O blood type, they don't have any antigen on the surface. Now, the white blood cells, the lymphocytes that they have, they can produce different antibodies. If it's the AB blood type, they don't produce any, right? There's no plasma antibodies because you don't want to go against A or B because they possess both of them, right? B blood type, they have an anti-A. Okay? That means you can't give it A. They have anti against A. So they can produce proteins to attack A blood type. A blood type, they have anti-B. And O blood type, they have anti-A and anti-B. All right. What that means is you can't give it the A, you cannot give it the B. All right. So it tells you blood type, who can receive what, and again, it gives you an idea of what's going on. All right, so let me further discuss this. As far as the RH group goes, again, there are different kinds, C, D, E, D are mostly common, but if they possess that, that's the one that your lab simulation or the labsters was mentioning. So, but if you possesses the RH antigen, then it's positive. If it's absent, it's negative. That's the easiest way to put it. Now, if you're a negative, you can give it to the person who's positive. If you're a positive, you cannot give it to the person who is negative. So in other words, they uh, going to have a problem because they're producing different antibodies and such. And again, your labs there went over this pretty well. I thought you have to administer ROGAM and the different type of antibody. Mother is, for example, certain blood types. The baby is certain blood types. During the pregnancy or giving birth, some of the blood gets of the baby, which is supposed to be a positive, gets introduced to the mother's blood, and then the second child, they can show antibodies. They can pass the placenta and attack the baby. Also, there are different type of uh, uh, antibodies too. I'm not going to go over different type. Again, Labster went over IgM, IgE, the different types. Okay, but we're not going to do that right now. I wanted to talk to you more about the principle rather than just individually. Antibodies are protein molecules. Protein molecules, they come from chain of amino acids. So if you have the DNA, you can make the messenger RNA, you can make the protein. All of that happens in the white blood cells. What type of white blood cells? Well, we mentioned lymphocytes, B lymphocytes that produce antibody. Okay, there are also plasma cells. They can turn into a plasma cell. They can produce antibody too. There are memory cells. Memory cells, again, they can produce antibody quicker, all right, on the exposure, second exposure. But anyways, chain of amino acids, usually, again, this is the heavy chain. This is the light chain, kind of. And there are disulfide bonds that connect them together, sulfur, they connect them together and so on. And most of the antibody, this region is similar. This is referred to as constant region. But over here might be different now. They're different shape. These different shape. Okay, depends on the antigen. So if you wanted to make it like triangular right here, all right, let's say our foreign cells have this particular antigen on the surface. This part may vary. They may attach to this one, epitope, or this is the variable region. Now you have another antibody, maybe a different shape. You see, it's hard to draw these antibodies like that to have the heavy chain and the light chain and be a specific. So for that reason, we could just simply draw a Y shape and then over here emphasize the variable region would be different. So again, you could have so many different ones. And these types are different, but the rest are this kind of like the same. You know what it reminds me of? It reminds me of those wrench and the sockets that you could change them to different size and different shape, but the base of it, where the handle is always the same. If you're not familiar with the tools and so on, don't worry about it. I'm just telling you that you could change the tip, for example, to a different one. Uh, uh, these are like kind of like tips because your screws might be different shape. What are the screws? Uh, Analogy of the screws are kind of like antigen, right? These are what you opens up the screws with, all right? So that could be a good example of it. But anyways, these are the antibody the body produces. If you have a foreign now antigen, usually are the protein molecules on the surface of bacteria, viruses, even different cells, red blood cells in our case, and they could evoke or elicit our immune response. That means our immune is going to react towards it and might attack this one. All right, so how do they work though? Well, they work several different ways. And we're going a little bit towards the immunology, but that's okay. You guys, hope that you're enjoying this. All right, so one way to do this is uh, by recognizing the antigen. So if they are here, let's say it's a bacteria. Bacteria have their own antigen. Huh? Bacteria uses this antigen, and viruses too, by the way, including the COVID-19. They can use this, uh, protein molecule or antigen, and they could use it as a landing gear, and they could penetrate. Now, once inside, then it could replicate to many more. You can use our space or nutrients or energy supply, whatever, they could multiply like crazy. And pretty soon the cell is going to get infected and then it lies and then now it could go and infect others. That's how viruses and sometimes bacteria works and occupy, but you'll have a lot of infection going on. So one of the best method is to prevent them from attaching to our cells. So in other words, the antibody can surround this, can surround all these points of attachment to the cells. When they are surrounded, then they cannot get in. That's one way. Another way is called agglutination or clumping, where uh, is more relevant to the blood typing too. So they have two arms minimum. Some of them, they have more arms, but in here, because they're attached to one another. But anyways, uh, in here, you could see one arm can attach to one, another one to another, and again, maybe this one to one bacterial, this one, or red blood cells for that matter, this one to another. So you see what happens. All of these now is stuck together. Think of these as a handcuffs when you're using the antibodies, and think about the bacteria as the bad people. You know, if a police officer is few, they cannot, they handle them. They have to handcuff them together. Then they have nowhere to go. They're all stuck to one another, right? So uh, these are the major two ways that is relevant to what we're going to be talking about. Eventually, the macrophages comes in and engulf this, all right, and pretty much destroy it, all right. Same thing in here. They engulf like a whole bunch of them all at the same time. See, here's a macrophages maybe coming in, and they are engulfing a whole bunch of them all at the same time. So again, it gives you an idea of how they are working. Okay, that was a good example of bacteria. What about the blood? So again, arbitrarily, we can go ahead and put the A blood type as what type of antigen do they have? They have antigen A. What does that mean? That, well, we could assign this shape for antigen A. All right. B blood type, they have antigen B. Again, we can use the same as you booked it. This would be antigen B. AB blood type, they have both. Not only they have A antigen, but also they have a B antigen. So they have both. O do not have any, no antigen on their surface. Okay. Now, next up is what can they produce? All right. In particular, lymphocytes, white blood cell could produce, if a person is A blood type, they produce anti-B. What that means is this type. All right. If they are a B blood type, they can produce anti-A. What that means is this type. That means if you give it the wrong blood type, A blood type, to a person who's B, they have anti-A. AB, none. They don't have anti-A or anti-B. Doesn't make sense because then it would be attacking itself. And O blood type, they have an anti-A or anti-B. What does that mean? That means you cannot give it to them. Anti means against. So understanding this is important because then it will tell you who can give who, all right, and who can receive from who. So we can put another title, can get from and can give to. So A blood type can get from O or A, all right? Why O? Because O doesn't have any antigen on their surface. This one has only anti-B. There is no anti-O because there is no antigen on the surface. So you cannot give it a B. What is remaining is O and A. O is actually universal donor, so O can give it to anybody. Right? B blood type can get from O, but they also can get it from B. A B blood type can get from all. They can get from A, they can get from B, they can get from AB. Why? Because they do not produce any antibody against any antigen. So they don't have any antibody production because they possess both. So this is called universal recipient. O blood type can only get it from O. Can give to. A can give it to A, it can give it to AB. B blood type can give to B or AB. AB blood type can give it to AB only. O blood type can give it to everyone. A, B, AB, and O. This is a universal donor. Now, let's go to the RH antigen or D antigen. Again, there are different kinds, so I'm just calling it RH antigen. If it's present, then it's positive. Right? If it's absent, then it's negative. So if you have an A blood type, you are using A antigen. This, if it has only A antigen, that'll be A negative. Right? A positive, not only they have A antigen on the surface, let's assign a square for RH antigen. Then this would be A positive. Okay, in general, negative person can give to positive, no problem. Positive person cannot give to the negative. That's a problem because if you're negative, like an A in here, they could possibility of producing anti-RH because they have against that. They don't have it. They may not recognize this RH antigen and they can attack it. Again, a mother who is negative but the child is positive is kind of like that, right? She builds up this anti-RH, they can attack the child uh, blood, right? And that is very dangerous because some of the antibodies are small enough to pass the placenta, so they can attack the child, right? That's the idea. Now, um, next up, we're going to go ahead and take a look at how would you know. Well, usually paramedics, they always have an O negative with them. Again, negative because you can give it to whether it's negative or positive. O is the universal donor. So you could, not knowing what kind of a blood type it is, you're safe. You have O negative in your hand, give them O negative. This is why O negative is pretty much wanted all the time, especially by the Red Cross and so on, because it's universal donor. All right. Now, um, however, what if you do not have the O negative? You want to know the blood type, or you wanted to, uh, you wanted to, sorry, if you wanted to know the person's blood type for whatever purposes, right? Right now, there's a big fuss about the coronavirus, and some scientists are studying to see how many people with certain blood types getting coronavirus with different symptoms. As you know, there are different strains. So if they want to know, you know, okay, this one had a mild symptoms, or this many percent of the population infected had a A blood type, B blood type, or O blood type, O negative, B positive, and so on, you have to get the patient's blood type. All right. So mm, that's what we're looking at. This is not the only reason, but this is just an example that is relevant to what's going on right now. Again, you could look it up to see if you find that answer and share it. But anyways, uh, interestingly enough, you need to do the blood type. Now, here you get the kind of like, it looks like a tray or microscope slides, right? And there are three parts to it. So let's draw three parts. One, You don't get to do this in the lab, but you could still look at it and you are responsible for it. You get to do this in the simulation. So in the simulation, each of these, they have antibody on it already. So you could just put in the different blood on here from the patient. Let's say this is my patient that got sick and I want to know what is the blood type of it, right? I don't know the blood type, all right? The Labster simulation, they already labeled these, I think if I recall correctly, I looked at it yesterday. Uh, so there are two ways. One is having the antibody coded already. So you label this anti-A, anti-B, and or anti-RH. So they are coded with the antibodies already. So now you have to do just get the blood sample and just put it in here and look for the reaction. All right. Uh, or you could put in the blood first. This is what we usually done in the past. Put the blood sample of the person, one drop in each one, and then label this as A, B, and RH, and then add the one that is labeled A, anti-A to it. This one labeled as B, you add anti-B. This one, you add anti-RH. Well, when we put anti-RH, what are we really adding? Anti-RH, we are adding antibody that looks like this. What about anti-B? Anti-B, we are adding antibody that looks like this. How about anti-A? We are adding antibody that looks like this. So now, picture this. Let's analyze the first one. Let's say you put in the drop and then you put in the anti-A. If this person, let me erase the top so you don't think it's all negative. Well, I can't erase it right now because I saved it. Okay. Let's say we don't know the blood. You're adding anti-A. You're adding this type to a blood. Now, if the blood, here's the RBC, have the antigen A on it, it looks like triangle, then the anti-A would surround this, and again another arm would attach to another RBC, and so on. So lots of these will show coagulation because they're all sticking to one another and they're all A blood type. So if this part show clumping or coagulation, clump or agglutination, so there's coagulation, agglutination, clumping, they all means the same thing. If they are stick to one another, usually what you see in here, maybe you could observe it online, okay? Because I cannot draw it like them, but they're gonna be like clumping, like, you know, clumping. The red blood cells are clumped together, okay? That's called agglutination. What if it's not? Then it's not. You know, you're just not gonna see any. It's more like, you know, it's not coagulating. It's more uniform. The color would be more uniform. That's what you're looking at. So if I'm adding anti-A and it shows clumping, that means it has A antigen on the surface. But does it have B? I have to try that. Look at this one too. Does it have positive or negative? I have to look at all of them. So if this one is uniform and this one is uniform, that means B and RH is uniform, then they don't have the RH. They don't have the B. Otherwise, it would have gotten coagulated or clumped. So my conclusion would be A negative in this case. All right. Now, what about B negative? What would the B negative look like on your slide? You can pause the video if you wanted to kind of like calculate it. A B negative would have only B uh, antigen on the surface. So which one do you think will coagulate? Hmm? Again, these are referred to adding anti-A. This one is adding anti-B. This one is adding anti-RH. So if you answered this one will show clumping, you were correct because it has antigen on the surface. When you're adding anti-B, is going to show. Doesn't have RH on the surface, so that would be negative. All right. What would be the AB positive look like? AB positive look like? AB positive, what does that mean? You can draw it for yourself. That means it has A antigen, it has B antigen, and it has RH antigen. RH is present, so I have to draw maybe squeeze the squares in here somewhere. So now, what would you see? A, B, RH. Again, A is porting anti-A, B porting anti-B, RH porting anti-B, or they stuck to it, whatever. That means they're all going to show agglutination. Why? Because they possess the antigen. Antibody is going to recognize the antigen, and they're going to coagulate. So that's how you could tell the blood type. Let me go ahead and ask you this time. I'm going to give you the result, and you tell me what do you think it is. Here is your slide. Here's the A, here's the B, here's the RH. Assume again, you're adding it anti-RH, you're adding anti-B, and you're adding the anti-A. Your observation. A, no clumping. B, no clumping. RH, no clumping. What is the blood type of the individual now? Pause the video. Think about it. Nothing clumped. If nothing clumped, well, let's analyze. Anti-A, when you pour in anti-A, didn't clump. That means it does not have, this is my red blood cell, if you want to draw it. That means it does not have antigen A on the surface. B didn't clump when I added anti-B against the B. That means it doesn't have those circle either. Anti-RH also is going to recognize the RH antigen. If it doesn't clump, that means it doesn't have it. So in this case, what do you have? If you got O negative, congratulations. I think you understanding the blood typing. All right. The genetics of blood typing, it's simple too. Remember, sometimes this antigen referred to as immunoglobulin. So the genetics of it is simple. You have genotype and you have phenotype. So the genotype is the genes that give rise to the phenotype. Phenotype is observable characteristics. So you have A, B, and O. We're not concerned about the RH right now, but the genotype for A, it would be IA, immunoglobulin A. Again, you're using two, one set of the gene comes from one parent, another set of the gene comes from a different parent. So for every characteristics, you get two copies, remember that. But also you could have IA and little I, all right? So these are multiple alleles. Alleles is an alternate form of a gene, all right? So this gives me A blood type. B blood type, you have this particular genotype or you have this particular genotype. That gives me B blood type. So remember the capital is was for or uppercase was for dominant characteristics. So what is that telling me, this one? Well, let's go ahead and take a look at O. O is little I, little I. That means it's recessive. That means if you get a gene or an allele from one parent that is A or B, take a look, they're dominant over the little I for the O. So what that means, if your mother is O, you will get one of these. Your father, let's say, is an A, you will get one of those. Let's say that this is your father genotype. Okay? Then you get this, you get the one gene from each parent. All right, so you get IA little I, which again translate to A blood type. AB, that would be AB genotype. All right. So the genetic part of it is actually pretty easy. You just have to figure out first how who is uh, the parent genotype, and then you'll be able to figure out what would be the offspring. All right. Now, when the two copies are exactly the same, this is called homozygous. When the two copies are different, this is called heterozygous. So this is a heterozygous. All right, so we discussed this already in the previous chapters or discussion, YouTube videos, whatever. So this is a heterozygous. So individual that is homozygous B, homozygous means they're both the same right here. Marries and mating or crossing, just put the X. With the individual that is uh, homozygous A, so this is a homozygous A. That means both of the copies is the same. What would be the offspring? Well, you have to get one from each one. One here, one here. You cannot take, two from one parent. The sperm carries half of the chromosomes. Egg carries half. So if this is male, this is the female. All right. The sperm carrying one of these is going to hit the egg that carrying one of these, and then you get AB, which that's the genotype. The phenotype would be AB. All right. So if you want to do a Punnett square, you could. You could draw 4 by 4 squares and separate the gametes. Gametes are sex cells. Sperm and eggs are referred to as gametes. So let's put the male on this side. Uh, IB and IB. I have to separate them because during the process of meiosis, if you have 46 chromosomes, you're going to get 23. This is 2. All right, we got to get 1. All right, so that means what? That means the sperm carrying a chromosome with this particular gene in it. All right, now with the female, we're going to put it this way. If you want to, we can draw a different color, but you could do different color if you want. For the female, it looks like IA, IA. This is the egg containing. this particular chromosomes with this particular gene. So sperm hits the egg, you write your results here. This sperm hits this egg, then you write your results here. I, A, I, B. A comes before the B, so that's why I'm doing it here. And so on. So if you do all of them, you'll notice like you're only going to get AB. There might be some genetic questions on the next exams, especially the ones that are related to the blood. So make sure you look at the X-linked hemophilia. You're looking at you look at X-linked recessive hemophilia and color blindness. You look at the sickle cell anemia, which is autosomal recessive, and you're looking at the ABO blood typing and how you could cross. So if you want to have more practices, you could. Individual that is heterozygous A, so heterozygous A would be here. Marries individual that is heterozygous B. There you go. That's heterozygous B. Notice that the A and B, they're both dominant over the little I allele that was from O. This is also known as co-dominant. Co-dominant, that means like co-worker. Together, they're both dominant. All right, so what do I do that? Well, how? Same thing. You could assume it really doesn't matter because they're not sex linked. It doesn't matter if you change the sex, but for arbitrary, this could be male, this could be female. And again, separate the gametes to put it in the sperm. I would draw the sperm, so showing that they each carry one of the uh, alleles or alternative form of a gene. And same thing with the female. I'm going to put it across. So here's the egg. The sperm hit the egg. I got to write my results right down here. This sperm hits this egg. I got to put my results right here. This sperm hits this egg. I got to put my results down here. And so on. So in this case, you can see we could get all different ones. You could get O blood type, you could get A blood type, you could get B blood type, you could get AB blood type. So in this combination, you could get any. In fact, it's like 25% chance of getting each one. 25% O, 25% A, 25% B, 25% AB. So that's how it works. All right. So if you want to know the phenotypes ratio or the genotype ratio, that's what they refer to. How many percentage you get AB, how many percentage you get A, B, O. All right. So you could have fun with it. You could try different ones. You could try different ones to see if, uh, you know, you could, you know, cross the A individual with O. All right, see what you get. Heterozygous A with O. You could try different combination and see what you get. Interestingly enough, if you know your blood type, you could ask your mom and dad if you want to. I don't know if you want to, but if you want to, I suggested this to the student, and the student was saying that's not a good idea because what if the person is adopted? You know, if you're adopted, then you this may not make sense to you. But anyways, if you know your mom's genotype uh, or phenotypes, you could guess. Now, if it's A, you don't know if this is it or this is it, but you could still try both of them to see which one you are. You know your blood type, you know your mom's blood type, you know your dad's blood type. You'll figure it out. Okay. So let's say that your blood type is B, but you're not sure. Is it B negative, B positive? You know, is it heterozygous B? Is it, homozygous B? Again, we're not worrying about the all the RH right now because that genetic of it is a little bit more lengthy and complicated, but let's just say you're a B, but you don't know which is your B genotype. Is it this or is it this? Now, your father is O blood type and your mom is B blood type. So if you're B, you must obtain the B from here. Again, your mom has two possibilities. It could either be this or it could be this. You only can get one of the allele, one of the genes. Your father is simple, it's only this. So you must have gotten this one from your father, all right? And the B must have gotten from the mother, all right? So this one was from the mother, this one, this is your genotype in this case, all right? Uh, it doesn't make sense to be homozygous. You must be heterozygous. So, anyways, you could have fun with it and try different things and go from there and try different combination. Same thing with the ABO. I might include some reactions, for example, for you to analyze on the exam. So make sure you Google it and look at it to see what would the reaction would look like, what is the coagulation is going to look like, or clumping, and what if it's transparent. So make sure you do that. Anyways, I will be posting this up for you guys. If you have any questions, you can ask me later. But at this point in time, I hope you guys enjoyed the lecture and learned something again. Uh, in hope of better days. Talk to you guys later.